Bismillahirrahmanirrahim. Assalamu alaikum everyone. I am Dr. Saba from Department of Microbiology and Molecular Genetics, the Women University Multan. And the title of topic that I am going to present today is bacterial biofilms and efficient absorbent material for arsenic remediation. This is the abstract of my paper that I am going to present today, but I will not completely describe this abstract over here because you already would have seen it in the abstract booklet of the conference. So let's start with introduction of the topic. Pollution of surface and groundwater bodies due to arsenic is one of the most important and life-threatening problems worldwide, with approximately 150 million people consuming arsenic-contaminated water. So this is a very large number of people, group of people that are consuming arsenic-contaminated water on the daily basis. And this arsenic is highly carcinogenic and poisonous toxic metalloid that is continuously going inside the human bodies, and we need to remediate this arsenic contaminated water. So let's see an approach of bioremediation. So bioremediation of arsenic through microbial biomass constitutes biosorption, that is an extremely efficient process due to low cost, reusable biomaterial and sorption of toxic metals and metalloids within short time periods. So this is the economic method and eco-friendly method by which we can remove the excess amount of arsenic from the contaminated water bodies. And one of the living methods is the use of EPS, that is extracellular polymeric substance, an essential component of bacterial biofilm. So let's see what is an EPS. It is a complex high molecular weight extracellular polymers that are secreted by microorganisms and mainly composed of hetero or homopolysaccharides, proteins, nucleic acids, lipids, and humic-like substances. So this is the composition of EPS, and different environmental factors such as carbohydrate sources, dissolved oxygen content, temperature, pH, metal ions, and shear forces, etc. may alter EPS composition, structure, as well as the properties. So EPS is affected by many different factors, and this may vary depending on the situations that are available to the bacteria in the surrounding environment. And EPS is generally present in two different forms that include soluble or colloidal EPS. It is excreted outside the cellular surfaces, and the second one is accumulated within microbial aggregates and tightly bound to the cells, known as bound or capsular EPS. So EPS produced during biofilm formation exerts several beneficial impacts on bacterial cells, including their protection from harsh environmental conditions like toxic metals and antibiotics, retention of water, and enzymatic activities. So this is basically biofilm formation is the protective action mechanism of bacterial cells to prevent them from harsh environmental conditions that may be in terms of some metals or in terms of some antibiotics or other harsh conditions. Moreover, changes induced by EPS in the bacterial cell surface, hydrophobicity, charge, and viscoelasticity help in cell adhesion and aggregation for biofilm formation. Bacterial EPS contains many different types of chemical compounds, predominantly acidic polysaccharides having ionizable functional groups like hydroxyl group, carboxyl, amide, sulfate, and phosphoryl groups that interact with toxic cationic metals and metalloids. These polymeric compounds present in EPS undergo electrostatic interactions with metal cations and therefore reduce their mobility or toxicity through accumulation or immobilization within EPS. So the EPS having different polymeric compounds actually immobilize toxic heavy metals inside themselves, and they serve as biosorbent material for removal of these toxic metals and metalloids from the environment. Immobilization of toxic metals and metalloids constitutes an important and promising strategy to alleviate the toxic metals from environment. Metabolism independent passive biosorption of toxic metals within EPS is more favorable than metabolism induced bioaccumulation inside the bacterial cells. Therefore, EPS has been investigated for the sequestration of toxic metals, including arsenic, due to its metal binding properties. So, this study was aimed to determine the role of EPS as a suitable absorbent material for arsenic bioremediation. So we wanted to target arsenic, and this study was designed to actually use bacterial biofilm EPS as a biosorbent material that is economical and eco-friendly for the removal of arsenic from our environmental sources. Therefore, effect of arsenic toxicity on bacterial EPS production, chemical composition, and structural characteristics was studied through different colorimetric quantitative methods, Fourier transform infrared spectroscopy, and scanning electron microscopy. So now we will discuss the methodology that was followed during this study. First of all, there was acquisition or selection of bacterial strains. In this study, for the purpose of bioremediation, we used two strains of arsenic-resistant and biofilm-forming bacteria. These bacteria were Exigobacterium profundum PT2 and Ochrobacterium ciceri SW1. These two strains were previously isolated from industrial wastewater effluents, and they have been characterized as arsenic resistant as well as biofilm forming bacterial isolates. Secondly, these bacterial strains were checked for their slime production assay by using Congo red agar method. In this, the ability of bacterial strains to produce slime was detected qualitatively by Congo red agar assay. Congo red agar plates were supplemented with and without arsenate or arsenite, and these plates were prepared and bacterial strains were inoculated on these plates, and after overnight incubation, the colonies were checked for the production of black color that will indicate the production of slime or biofilm on the surface of Congo red agar medium. Thirdly, the bacterial biofilms were grown and their EPS was extracted by following the method in which bacterial pellets was treated with ethanol, and it was under the cold press method. Then morphological and biochemical characterization of EPS was performed, and in this method, EPS was extracted, and after extracting, its color, texture, and weight was recorded, and protein content was measured by Bradford assay, whereas total carbohydrate content was measured by using colorimetric phenol sulfuric acid method. Moreover, morphological characterization of EPS was also done through scanning electron microscopy, and scanning electron microscopy images helped us to identify different morphological changes that happened in EPS due to the stress of arsenate and arsenite. Moreover, surface characterization of EPS was also done through Fourier transform infrared spectroscopic method that helped us to identify different functional groups that were affected due to the stress present in the form of arsenate and arsenite. Now let's move towards the results. As we checked the slime production assay for both bacterial strains that were selected, it was shown that Exigobacterium profundum PT2 displayed slime production in the form of black colored colonies, both in the presence and absence of arsenic. So their slime production ability was not affected by arsenic stress. However, in the case of Ochrobacterium ciceri SW1, it did not produce black colored colonies on Congo red agar plates, neither in the presence nor in the absence of arsenic stress. So, uh, Ochrobacterium ciceri was unable to produce slime. Here you can see the red colored colonies that are indicating negative results for slime production, whereas these black colored colonies are showing positive results for the slime production. Now comes the morphological and biochemical characterization of EPS. If we talk about the morphological features and quantification of the EPS produced under arsenic stress, so let's see that in the case of Exigobacterium profundum PT2 bacteria, brownish powder was produced under control conditions. However, it turned into whitish powder under arsenate stress and grayish powder under arsenite stress. Whereas the quantity of EPS produced was maximum in the presence of arsenate stress, followed by in the arsenite, and minimum amount was produced in the control when there was no arsenic present in the growth medium. However, in the case of Ochrobacterium ciceri SW1, control strain also produced EPS that was less than that produced in the arsenate and arsenite stress. Now let's see carbohydrate and protein contents of EPS extracted from Azygobacterium profundum and Ochrobacterium caesari. So carbohydrate content was maximum in the presence of arsenate stress and then in the arsenite, and minimum was in the control condition, and similar trend was observed in the case of Ochrobacterium caesari. However, in the case of protein, the maximum amount of protein was observed in the presence of arsenite, followed by that in arsenate, and then control condition. And similarly, in Ochrobacterium caesari, maximum amount of protein was seen in arsenite, then in arsenate, and then in control condition. So we can say that protein concentration was increased under arsenite stress, whereas carbohydrate concentration in the EPS was increased under arsenate stress. This is the scanning electron micrograph of EPS produced by Azygobacterium profundum PT2 under arsenate and arsenite stress conditions. And we can see that in the control conditions, the biofilm EPS was having pores-like structures. These are pores when these are magnified to higher resolution. However, in the case of arsenate stress conditions, you can see that there are very large agglomerations and clumpings together. These are actually enhanced amount of biofilm and enhanced amount of EPS in the picture, that is showing the accumulation of different carbohydrates that we have also seen in the quantitative method, that carbohydrate content was increased under arsenate stress. So that is also evident from here in this picture. And now if we come towards arsenite stress, so the biofilm, the EPS is again very enhanced, but here EPS is having different structure, that is of proteins, like because we also run control samples of proteins and sugars along with these samples. So we compared them with the literature, and this is evident that protein content is high in the arsenite, whereas carbohydrate content is high in the arsenate stress conditions. These are the results of FTIR of Azygobacterium profundum PT2. And if we see this graph, then the peaks between the range of 800 to 900 per centimeter wave number, they are showing the presence of different glycosidic linkages. And in the control, the peak around 1211, whereas in the arsenate and arsenite treated, the peaks of 1218 and here 1134. These are also representing the polysaccharides and increased production in the arsenite as compared to control. So when we see the peaks in the range of 1700 per centimeter wave number, so these peaks are actually showing the presence of amide groups. And now we see the spectrum, FTIR spectrum of Ochrobacterium caesari SW1. So it peaks also around 847, 800, and 800, 847, and 880. These are showing the presence of glycosidic linkages. So we can clearly see that arsenate, the EPS of arsenate is having highest concentrations of all the functional groups present inside the bacterial EPS, whereas that of arsenite is less than arsenate, but still higher than control conditions. So in this study we found that EPS produced by biofilms of both Azygobacterium profundum and Ochrobacterium caesari, they served as excellent biosorbent material for arsenic remediation, and we can further more characterize these bacterial strains and use them for some practical approach to remediate arsenic from the environment, and devise some biofilm based biofilter that will be used to treat water and remove arsenic from it. So this was all about the topic that I presented today. If you have any questions, you may ask. Thank you so much for your precious time and for listening me.